Well, good evening and welcome. It's wonderful to see all of you here tonight. I'm Dorothy Harrell, and I'm the Chancellor of the University of Colorado, Denver. And tonight is a really special night because it is the inaugural Faculty Distinguished Lecture. And many of you may be aware that we've had here at CU Denver a Chancellor's Distinguished Lecture for a number of years. And that was made possible by Drs. Lenny and Robert Damrauer, who are here. And Bob and Lenny, thank you for the inspiration that then said to us it was really appropriate for us to establish a lecture that featured one of our own faculty members. And part of what makes CU Denver a really special place is that we have the distinction of being the only public urban research university in the state of Colorado. And so finding opportunities for us to elevate the really important scholarly and research work that our faculty do is something that we're paying a lot of attention to. And this lecture, it will be the first of an annual lecture, is going to be one of the keystones of our efforts to do that. So our speaker tonight rose to the top from an internal competition, that there were many talented people who were nominated, talented faculty members throughout the university. And I think we have the three finalists that emerged from that process here tonight, and I'd like to congratulate and introduce them as well. So we have Professor Jim Grigsby, who teaches psychology in the College of Liberal Arts and Sciences. Jim, are you here? There he is. Jim, thank you. Professor Diana Tombeck, and Diana teaches integrative biology in the College of Liberal Arts and Sciences. Diana. And our third finalist, Professor Ron Suhr, who teaches mathematics education in School of Education and Human Development. Thank you. So our lecturer tonight is Dr. Stephanie Santorico, and she is a professor of mathematical and statistical sciences. That's a mouthful. She's been at CU Denver for 11 years. And one of the things that Stephanie says is that she's passionate about the interface between science and statistics. And she finds that the best statistical methods come out of collaboration, ensuring that new discoveries are meaningful contributions of knowledge. She brings a background of high-impact work, including being among the first to study genetic influences on gene expression in humans. She's currently working on methods for genetic risk prediction and disease subtyping. Beyond being passionate about research, she places great importance on the teaching mission and believes she best serves our students by striving to conduct meaningful research and using this experience to contextualize the course material. I was interested to know that some of her work, she partners with the Denver Crime Lab, where she's developing new methods to catch criminals by using partial DNA matches of their relatives. And she offers this advice to aspiring researchers. And I think this is probably the advice that she followed herself when I think about it. She says, find an area that you are passionate about, make a plan, develop a network of mentors, then move stubbornly toward turning your passion into long-term growth and knowledge with lasting effects on your chosen field. She also asked me to mention that she's going to be talking about a few companies in her presentation this evening. And she is not an employee of those companies. She doesn't own stock in those companies either. So we can be assured that what she's going to be sharing with us is totally unbiased. So another sign of a wonderful researcher. So please, will you join me in giving a very warm welcome to our first faculty distinguished lecturer, Dr. Stephanie Santorico.
Okay, thank you for that kind introduction. And thank you all for coming on a Wednesday night to learn a little bit about genetics and how they apply to you. So hopefully you managed to get a handout on your way in. We're going to use that for a number of things, so if you didn't, make sure you get one. You'll see there are a few spots on there where I'm going to have some prompts for you to be thinking about and writing some stuff down. We are going to be defining different terms, so there's going to be terminology. Just in case you forget some of those things, because I'm going to be giving you a number of them, I give you key definitions, so you can always refer back to that. Maybe for you. And we've got some more coming around here. Just to keep you lively and participating, at the bottom, there's a fill in the blank. So I'm going to be giving you some facts, right? So besides me thinking that these are interesting facts, these might come in handy at trivia night, so you can impress your friends and family. Anytime you see the little handwriting symbol on the screen, that's a clue that a fact's coming up. On the back, a couple of things. So I provide references to a few of the things I talk about. I provide some web resources for you. So if you're interested, and I hope you are inspired, you've got some links for following up. And then possibly even more importantly, at the end, I have an area for questions. And you're certainly going to have questions. Please, as soon as they come down, just write them down, jot them down. We're going to have a Q&A section at the end, and so hopefully we can get to your questions then. So with that, let's talk about O.J. Simpson, right? So O.J. Simpson and the one degree of separation between him and my research career, right? So it was 1994, and if you are of at least a certain age, you will remember much of the nation glued to their TVs watching this chase. Right? It was one of the first times I think we really saw that on TV. If you are not of that age, you probably saw it on the Netflix series. So 1994, here we go. About a year later, my soon-to-be PhD advisor provided testimony at the trial. And so what I saw was somebody who loved math, just like me, using statistics to interpret genetic data. Right? So the first time I'd really seen this. And I was always a nerdy little math kid. Right? So just to give you context, my favorite toy growing up was a little Professor Calculator by Texas Instruments. If you Google it, it's fun to look it up. Perhaps that should have been foreshadowing for my mother on my future career, but I loved that toy. So I always loved math. But seeing somebody use math and statistics on real data, right? So seeing them being able to use statistics to really take the bridge between mathematics to meaning was what sold me, right? So yes, O.J. Simpson. 2 years later, I am headlong into research, learning about statistics, learning about genetics. And that's where I started. Fortunately for me, along the same time, we had a data revolution that was beginning in genetics. So at the time of my schooling, the kind of data, and we still thought it was a lot of data at that point, we were routinely looking at around 400 spots of the genome. Right, that's still a lot, especially for someone coming in. I was a first-generation college student. I'd never seen anything that looked like this. 400 spots, that's a lot of data. Very shortly after that, so fast forward up to 2005, we were able to look at 100,000 different places in our genome and ask questions like, which parts of our genome affect a person's disease risk? Right, seems like a lot. This escalates very quickly. So by 2010, 500,000, shortly thereafter, 2.5 million, and now we are at over 3 billion places that we can measure and ask questions like, where in your genome is important for your particular risk of disease? Right? So we are able to look at human health at an unprecedented level. It's been ramping up, and there are so many open questions now, and we've got so much data to do this with. Before I get too excited and get too deep into this, I'm going to give you a few basics just to make sure we're all speaking the same language. So DNA, right? DNA, you can think of as your instruction manual, right? That's what tells your cells how to function, how to respond to your environment, and essentially how to be you. Just like the letters in the words that make up an instruction manual, DNA also has letters, but instead of 26, it only has 4. So A, T, C, and G, these are the abbreviations for the bases that make up your DNA. Right? How many? We have over 3 billion of those. So if you've seen DNA, you've probably seen it represented as this double helix or as a ladder. The rungs in that ladder are these 3 billion places. So our letters, A, T, C, and G, which make up the instructions that make you you. So a few more facts before we get a little heavier into disease, which is not the most lively topic to be talking about. If you took one of your cells, so just one, we've got lots of cells, and you took that DNA and you stretched it out, right? You can think cells are pretty small. If I stretch out that DNA, it's probably not going to be that big of a distance. It's actually 3 meters long, right? So 3 meters, one cell, 3 meters. To give you some context, that is the distance from the court to the net of a basketball goal, right? One cell. So let's take this a little bit further. So more trivia to impress your friends and family. So all of the cells in your body. So imagine your body, it's not that big. It's carrying all of those instructions and all of your cells. Take the DNA out end to end. Imagine how long that might be if one cell was 3 meters long, right? So what kind of distance are we talking about? We're talking about the distance from the earth to the sun and back, not just once, 70 times. 70 trips from the earth to the sun and back. That's the amount of information that you are carrying in the cells that you have in your body, right? An amazing amount of information. That said, if we were to look person to person, we are actually mostly the same, right? In fact, so if I were to represent a person as a pie, because we all like pies, this blue area is all of our genome where we are the same. So any of the pairs of individuals in this room, we're the same at 99.9% of our genome, right? It is that little bitty sliver which you can barely see, kind of an orange little line there. It's those 3 million places that make up the amazing diversity that we see in the human species. Right, so 3 million. And what scientists have been doing over the past few decades is looking at those 3 million places and trying to understand which places are important for which health and disease traits. So that's what we're focusing on. To put this a little more in context, what I would like you to do, well, first, a favorite quote of mine. So one of my favorite quotes is Maya Angelou. I said I was a nerdy little math kid. So reimagining this instead with a nerdy twist, and specifically in the lens of genetics, we are indeed vastly more alike, right? How much alike? 99.9% alike. Right? So we only differ at a very small sliver of our genomes. So I'm going to ask you to do something. So I'm going to give you a minute. And what I would like you to do is introduce yourself to your neighbor, preferably someone you haven't met before. You guys are going to look different, but think about how much you really share in common. And you're going to hear a school bell, which will bring you back in. right. So thank you. So number one, it's late. It's good to kind of loosen up a little bit. It's good to meet people. But despite our differences, 99.9% alike with the person you were shaking hands with. So it's pretty amazing. So what we're going to do next is talk about our genes, and to give you some context for how we think about that little bitty piece where we actually differ. So our genome has segments along it, which are our genes. And in particular, humans have around 20,000 genes. Okay, so 20,000 genes located within our genomes. And the way that scientists look at this is we look at the spots, right? The spots where we're all the same, not that interesting. But we're interested in looking at the spots where we differ to understand how those differences might lead to a different risk for a person's disease. So some terminology. So the place where we're looking can have different bases at it. And these are called alleles. So alleles are just the different material that can occur at that location you're looking at. We talk about these in terms of the allele you got from mom and the allele you got from dad, right? These together, my parents were superheroes. So these together make up what we call a genotype. So if you have done an ancestry type test, your data is going to look like this. So for me, my genotype at this particular location is a GG. I got a G from mom, I got a G from dad. We call these locations specific names. So this one, which I'm going to spend a little bit of time talking about through the talk, is called RS4977574. It tells you nothing. It is not at all intuitive, but we use these names so that if I tell you what my genotype is at that location, you know what I'm talking about, right? So standard names. And if you want to, you can Google it and you'll know the same thing that I'm talking about, right? So we use names for these things. So this is part of my DNA, right? And specifically, I'm not showing you where we're similar. I'm going to show you a part of the places in my genome where I differ from you guys, right? So this is part of it. Looks like a lot of data. If I were to show you all of my data for all 3 million of those places, it would take me over 800 slides, okay? We're not going to do that. Scientists do not look at data through PowerPoint, so don't worry about that. What we do and what we've been doing for the past few decades is we look at one place at a time. So that one marker I showed you, I'm going to talk about. So we might look at that one place and study it. So how does that one place in our genome affect our disease risk? So genetics is very personal. So when I started thinking about how do I explain genetics and how it affects you, I instead took a personal approach and wanted to explain how it affects me. And so I'd like to introduce you to my little brother. This is Brandon. And Brandon passed away at age 27, right, from a heart attack. So that is incredibly young. Being devastated, of course, one of the things that kind of paralleling some of my training that I wondered about was, is there something in his genetics that maybe if we had known, that we would have known he was at risk for a heart attack, right? And he's my brother, so we share on average, so another fact if you want an extra one, we share on average 50% of our genetics, right? So if he had something, maybe I have that too, right? So one of the things I did when I first started looking at this was I asked the question, is there something in my genetics that might predispose me to heart disease? Another exercise. So it is a very personal thing when you start talking about genetics. And so one of my recommendations for you is to start by thinking about 1 to 3, right? So I'll talk about my at least top 2, and anyone who really wants to know, I'll tell you my top 3. But what are the things that affect you? So diseases, traits, what affects your family? And you'll find a spot on your handout. Write down 1 to 3 things that affect you. And I'll bring you back together with the bell again. that one was a little easier possibly to come back from. So you got your top 3. So the question then is, how would a scientist interested in one of your top 3 look at your genetics to see which pieces of your genome are important for that trait? So I'm going to walk you through the way that we do this. Right? We're going to do it a couple of ways. So I'm going to kind of show you under the hood so you can see this is the kind of data and this is the kind of thing that scientists use. Right? And then I'm going to show you, and this is what it's going to be given to you as. Right? So we're going to go both ways. So for most diseases, what we do is we start by collecting a whole bunch of people. Right? So my top one was heart disease. And so for a study of heart disease, we would collect a large number of individuals who had heart disease and a large number of individuals who did not. Right? So healthy, we call them controls often. And we, for the past few decades, have been looking location by location in your genome. Right? So at one specific location, we would look at what are the genotypes for all of these people. Right? So the allele you got from mom, the allele you got from dad, and I want to know everybody's genotype. And lucky for me, there are all sorts of fancy statistics and fancy mathematical models that we can use to do this. But at the end of the day, all we're doing is asking the question, if I look at the diseased people, so if I look at people who have heart disease, are there differences in their genotypes compared to healthy individuals? Right? So if you kind of eyeball that a little bit, you might notice it kind of looks like the G allele is showing more over here than it is among the normal individuals. Right? This is just a picture representation. I'm going to show you what a real study looks like. Right, so this is data from a study. I've given you the reference to a paper in 2018. And what they did was collect around 35,000 different individuals who had coronary artery disease. They looked at roughly 260,000 people who did not. And you'll see that very descriptive name pop up again. So they got the genotypes for that specific location in a person's genome. And what the data here is represented as is for individuals who have disease, you just count. How many times did we see a G? How many times did we see an A? For the healthy individuals, same thing, just count. How many alleles are you seeing in the healthy individuals? Right? So this is the data that we're using. Then you can talk in terms of frequency. So if I were to estimate how often does G show up in diseased individuals, it's a 0.13 versus a 0.11 in controls. Right? So I can tell you, since I do statistics, that difference is highly unlikely to occur by chance. Right? It's a legitimate difference. Right? And this is what we consider a significant scientific finding. Right? You might not be sold by this. It doesn't look like that big of a deal, right? This is the data that we're using to make these statements. It's kind of a big deal because it at least points us to a gene and a place in the gene where we know this has something to do with disease risk, right? So biologically, still meaningful. Now, to the consumer, the way this data tends to get reported is with a whole different thing, a statistic. They're not going to show you these frequencies. They're going to give you something which, if you don't understand, can kind of be stressful. What they give you is something called the relative risk. Okay, so warning, a formula is coming up, and we are going to walk through it. The relative risk tells, so on the top is the probability of disease for you. So given your genotype, what is the probability of you developing that disease? It's relative to a baseline group, to a reference group, right? And typically what we do is we look at the most common allele in the population. Things that are more common are older, so we think of them as kind of being the normal type. And so the reference group is individuals that are just carrying 2 copies of that major allele, right? So this is going to be something that you would see looking at 23andMe results. Interpreted, all that means is if you have a value that's bigger than 1, right? So in the red area, that means you're more at risk than the baseline reference population. If you have something down below here in green, that means you're actually protected. You're better off based on your genotype. So for me, the location that I had been talking about, based on my genotype, so I told you guys I had a G and a G from mom and dad, my relative risk was 1.5, right? So 50% higher than the reference group population, right? 50%. It's kind of a big deal. If something's on sale for 50%, you're excited. It's a big quantity, right? But one of the things I want to make sure you understand, besides that definition, which is helpful, is this is always relative to a specific group, right? So it's relative. Let me tell you a little bit more about what that means. So for most diseases, so if in your top 3 you happen to write down some diseases in there, most likely that disease risk is going to be related to your racial group, to your age, and to your sex, right? It's relative. So for me, being mostly European of descent, being female, being my age, we're not going to get into that, my baseline risk is around 5%, right? So it's 50% higher than that, though, as soon as I'm told my genotype, right? So my risk of developing coronary artery disease, just based on the information at one location in my genome, is a little bit over 7%, right? So higher, but it didn't seem as higher as when you're thinking about 50%, really, because it's relative, right? So something to remember. We're going to get into this a little bit more, but it's not just relative to your particular demographics. It also is going to depend on a lot of other things. So for coronary artery disease, about half of risk is due to environment. So environment is things like your nutrition, your exercise, your blood pressure, your weight, right? So these environmental factors are going to affect 50% of my risk. Genetics is responsible for the other half of that pie, right? And I looked at one location, but that one location, okay, and this is what we've been doing for a couple of decades now. This is what 23andMe is using to tell you about your results. They are taking the view of this one little sliver of that pie to inform your risk, right? So it's all going to be relative. So just to kind of show you the amount of information we now have. So we've done this point by point for thousands of different traits. I'm going to show you a little animation, a little video. And what you're going to see is your chromosomes laid out. And as scientists found a location in the genome that related to your disease or to a health trait, you're going to see a little bubble pop up. Right? So I'm going to start that here. So just to show you, this is the science that companies like 23andMe are using to interpret your results. The web address is on your handout. So this is around 5,000 studies, and there we go. So starting from 2005. You can see all of the little dots starting to populate across the chromosomes. 2004, 15, all the way up to April 18th. So that was when they updated your catalog. Notice that we have so many different findings that they don't even line up with the chromosome. They're just completely, they have to be expanded for you to even see them. We have in this catalog, so my guess is if you looked at your top 3 and you went on that web address, you'd be able to find some of these findings specific to your particular trait, right? So that's a good resource if you're interested in digging in a little bit more. We have 70,000, they call them associations. And all this means is we have 70,000 places where we've been able to say this place in a person's genome is related to this trait, right? So 70,000 pieces of information like that. And this is what we are using. So this is a macro view because it's stacking in all sorts of different diseases. We've done this for certainly cardiovascular disease. We've done this for digestive disorders. We've done this for response to drugs. We've done this for cancer. We've even done this for just measurements like height and weight, right? So we have all sorts of things that we've looked at. But kind of digging in, so taking a micro view, we're going to look, because I'm talking and my top number one was heart disease, we're going to look at cardiovascular disease. So if you go in there, what they will tell you is there are roughly 1,000 places. So all of these little red dots are places in the genome that have been found to associate with risk for cardiovascular disease, right? I showed you one. They're saying there are at least 1,000. Right? That's a lot of different places. And so what I wanted to do is show you what this might look like, right? So if we recognize that one location is not enough, there's lots and lots and lots of locations that are important, then what we need to do, and what scientists are doing right now, is trying to combine information over all of those places, right? So think back to the pie, right? The pie was half orange. I showed you one sliver. What I really need to know is all of the rest of the slivers of that pie to put together to understand my disease risk. So we're going to visualize this a little bit. So this says, so that we can see it, I pulled 40. So these are 40 places in the genome. There are those super nice descriptive names right there. And then right beside them are my genotypes. Okay. We don't generally like to look at data in a table. So instead, we're going to look at a pretty picture. So what this is doing is giving you that relative risk. Okay. But we're talking about me, because I'm talking. So my relative risk is shown over those 40 different places. Right? So I've got bars for all of those genotypes. And what this is telling me, so if I'm above the bar, so at those particular locations, I have an increased risk for cardiovascular disease. For any of the locations in the genome where I have a blue bar, I actually am protective. So my genotype provides me a protective effect against cardiovascular disease. So the important thing to now notice is I told you we've been doing it place to place, right? So 2 decades worth of looking spot to spot. So depending on where we looked, so if I started somewhere like that, and what that told me was, oh, I'm at an increased risk for heart disease. Maybe I should work out a little more, or maybe I should eat more vegetables. Maybe I should be a little more careful because I have increased risk. But if we had started out at a place like that, that would have said, no, actually you're good. You know, skip the gym, sleep in. You got a good genotype. You're protected, right? So if you log on and look at just one place, right, you're going to get a changing story because these places have different kinds of effects on your disease risk. And so this is where we are now, right? So we've got all of these great resources where we're starting to understand places that do correlate with risk. But what we really need to do is look at them not individually, but look at them in sum, right? And so this is current research. So during the introduction, thank you, one of the things that I do work on is research into genetic risk scores, and with the idea of maybe identifying subtypes of disease, right? So we are working on this right now. Kind of the basic overview you can think about is we want to know, so if I take this risk and sum it up, right, a totally rough, not the best way to do this would be, do I have as many bars up as down, right? Am I balancing out, right? That's a simple view. Lucky for me, we don't know quite the best way to do this, so we're using statistics, we're using mathematics to try to understand how do we best come up with the formula that is going to tell me what my risk is. Right, so we're doing this now. We still have a lot of questions, right? So we don't know how reliable this is. I'm going to give you all the caveats for just a second. So we're doing it. We don't know how reliable it is. It's debatable how accurate it is from population to population. So if I've defined this using individuals in Europe, can I apply that same score to individuals in Mexico, right? So we don't know the answers to these questions, but we're working on it. That said, just because we don't know for sure doesn't mean companies aren't going to do it, right? So 23andMe, they did not have heart disease as a report, but they do provide risk for type 2 diabetes, related. So you share a lot of the same risk factors between those two diseases. And so I go on, I see, oh, they have this report for type 2 diabetes. Well, I have diabetes in my family. Maybe I should look at this. I log on and it tells me at my age, which we're still not talking about, I have a 35% chance of developing type 2 diabetes. It's big, like over 1 out of 3, right? They're doing this no longer on one particular place in the genome. This information is coming from them looking at over 1,100 different places. So looking at all of my genotypes at all of those 1,100 places, trying to fill in that pie, so get more of that orange piece, and using that to say, oh, this is your disease risk. Right? So I said a few of the caveats already, but since I'm talking, I might be trying to sell you something. I went to the news and I pulled some headlines, just so you're gonna believe me. So just from the last month and last month, a few representative headlines. Right, so things like, is the genetic test for diabetes risk? Why is it not ready for primetime care? Maybe they're not ready for the NHS, so maybe they're not ready to be implemented in the UK. They have a risk, a multi-gene test, that's the same thing as summing over all those locations, to predict depression risk, right? We're still trying to do this. We still don't know how well this is gonna work, okay? So something to keep in mind, cutting edge research. Regardless of all of those caveats, so regardless of whether we need to work on accuracy, whether we need to understand how well it applies to different populations, it's still all going to be relative, right? So relative to a term, so here's one of your definitions, to something called heritability. So heritability is the proportion of risk that is due to genetics. So genetics in orange, we've already actually been talking about that because that pie was part orange, part blue. The orange part is what corresponds to your heritability. So if we have a low heritability, so this is an example for hearing loss, so there much less of a person's risk is due to their genetics than environment, roughly 20%. Right? So regardless of how well we do on developing a score based on a person's genetics, we're only going to be able to explain this much of that pie. Right? We've been talking about coronary artery disease, heart disease. Type 2 diabetes is along the same. So about half of risk is environment, half of risk is genetics. And to show you the other end of the spectrum, if you look at autism spectrum disorder, over 90% of a person's risk is due to their genetics. Right? So these are opportunities we might have to do really well with a risk score because the biggest bulk of a person's risk is being driven by their genetics. So I'm going to show you a crazy figure next, so you're warned. I'm going to walk you through it. But perhaps more important, I put this address and the reference down on your handout. This was a study that looked at 150 different disease traits and estimated their heritability, right? So my guess is if you look at your top 3 and you're interested in, hey, how much of my risk is due to genetics, how much is environment, this paper would be a place to go to get an idea of that. And to give you a little walkthrough of the figure, so we have a spiral starting on the inside, and these are places where we have very small heritability, right? Meaning more of a person's risk is driven by environment. And we're spiraling out, getting higher and higher in terms of heritability. We break over half up here. So the type 2 diabetes, the heart disease is happening up there. And we go all the way out to some of the higher heritability traits. I already mentioned autism spectrum disorder, but things like ADHD, where 76% of a person's risk is due to their genetics. Glaucoma, aneurysm, bipolar disorder, lupus. These are all things where genetics plays a more important role than environment. And just to make sure you get the other end of the spectrum, perhaps not surprisingly, things like hernia, right? I already mentioned hearing loss, an eye infection, right? So those are way more driven by environment than genetics. So we're only going to be able to tell you so much from your genetics for those particular diseases. So back to my example, right? So 23andMe tells me my risk for type 2 diabetes based on 1,100 different places is around 35%. I was really amused to see they also provide a questionnaire you can fill out. On the questionnaire, they ask things like height, weight, and I was even more glad to see they ask about frequency of fast food usage and frequency of exercise. All right. So they actually take that and they are doing this is where we are heading. They're taking my 1,100 pieces that explain my genetics. They're adding on those pieces that get to my environment. And they tell me, oh, hey, your risk is under 1%. Right? So that message of it's all relative, right? So the fact that 50% of the pie, that environment, a lot of the things that I can control, I can use those to drive down my risk compared to just looking at genetics, right? So it's all going to be relative in terms of how we can do this. So this brings us back to our worksheet. What I'd like you to do is you'll notice right beside your top 3 traits, I left you a spot. So think about the different environmental factors. I'll give you about a minute and jot those down. Doesn't want to quit. So hopefully that'll give you a few ideas, right? So for me, when I looked at my top 1, there were certainly a lot of environmental factors that I control, right? So I love to exercise. I have a great community and network for fitness. That is a way that I can modify my disease risk. I'm hoping you kind of looking at yours, that might be similar for you. What are things you can do that from the day to day are going to have a large impact on your risk, right, beyond your genetics? And so it's going to all be relative. So we are going to go on to another personal story. Not as depressing this time. So I talked about the top 1, so that was heart disease, something I think about. My second one actually was hair color, right? So specifically red hair. If you are lucky enough to know a redhead, we are very proud of our hair color, generally, and even the stereotypes that frequently go along with it. But the reason that this is being brought up right now is because when we start to learn genetics, one of the first things we're told is genetics of hair color, genetics of eye color. Right? We're told these because these are, quote, simple traits. Right? So some foreshadowing there. A little bit of biology. So red hair is a recessive trait, which means you need to get the version of the red hair gene from your mom, the version from your dad, and those two together develop red hair. Okay? Possibly way more information than you want, but because a redhead is telling the story. The gene, so what you're told, the gene that is responsible for red hair is the melanocortin-1 receptor. And so that we do not have to say that again, we're going to use the abbreviation MC1R. All right, so I have red hair. My mom and dad both had brown hair. they must have been carrying the red hair version of that gene, but they must have also had the brown hair. Okay, so I'm going to show you the inheritance pattern for this. If you have seen this before, great, this is a Punnett square, but let me walk you through it. So what a Punnett square does is it says, I'm going to take, here's mom. These are the two different alleles that make up her genotype. And she could send either one of those to her kids. And equally likely, I can pass either one of those along. My dad also had brown hair, so I'm assuming he had that red hair gene since I have red hair. He's got the allele for the brown hair and the allele for the red hair. Okay? So what this would tell me, so what I learned in basic genetics, basic biology, is 3 of us, and there were 4 kids. So I was set up to go into genetics. 4 kids, 3 of us, so my 2 brothers and my sister all had brown hair. Okay? The 1 out of 4 is me. All right, so matches, the distribution matches, great. Me thinking as a scientist, it's always good to have a sanity check. So I log on to 23andMe and I pull up red hair color. Right, so a good sanity check. I'm told this is simple. I know what my genotype should be. And this is what 23andMe tells me. Granted, I can tell you I'm a redhead. All right, more and more of it, I have lost track of how much, but more and more of it is gray. But I promise you, I am actually a redhead. They give you a little more data, right? Great data junkie, give me data. So they tell me 3 markers, 3 locations in that gene, and they say, here are your genotypes. So at none of the 3 locations that they tested do I have the genotype for red hair. Right? Again, being a data junkie, I dig a little bit more. I found 6 other places in that gene, which have also been shown to lead to red hair. I pull my genotypes out. I don't have the genotype for red hair at any of those. Okay? All right. Interesting. So granted, maybe there's something else in that gene that they didn't type, so that's a possibility. Though I looked at 9 things, so the 9 most common things that cause red hair, and I have none of them. So I'm going to show you an article. So this is also on your handout, and this was just from December, so just 4 months ago, right? And what they say is it turns out that hair color is not as simple as we thought, right? So MC1R does account for a lot of redheads, but they actually give 8 more genes. So there are 8 other genes, so other places in the genome that put a person at risk for red hair. And an interesting thing is they actually then said, and hey, actually only 73% of the risk of red hair is due to that MC1R gene. Right? So a little over a quarter is due to something else, right? So what that also means, pun coming, MC1R does not explain all of the heritability of red hair. I had to throw that one in there. So why am I telling you this story? Besides being a redhead, loving my red hair, I'm telling you this story because if we don't understand and have the complete story for something as simple as red hair, right? So we still are not right on that. How much uncertainty and how much more do we have yet to learn when it comes to something more complex like obesity or autism or type 2 diabetes or heart disease, or my guess, the things that are on your top 3, right? So we've got a long way to go still in figuring these things out. That is, we are still learning, right? So we are still trying to contextualize lots of things. We can measure our genetics at an unprecedented level. So we're now starting to learn things we didn't know, right? So we have a long way to go. This said, all right, so if you start digging into your genetics and something gives you anxiety or makes you panic, heed my warning, stop, right? So we don't know everything. We could be wrong. We don't know how this applies to you and your context, right? I've been doing this kind of stuff for a couple of decades. I logged on and I looked at my late onset Alzheimer's risk. I looked at my Parkinson's risk. If I had been told I was at an increased risk for either of those, I would go talk to somebody, right? So I would, statistics, I love the statistics, but as soon as they apply to you, it means something different. So if you do encounter that situation, a great resource is the National Society of Genetic Counselors, and I have their web address on your handout. There are educational resources, and there are also ways to get in contact with a genetic counselor. So heads up, if you need it, go get it. If I would need it, I'm going to trust you might need it as well. All right, so one more biology lesson. So when we go through biology, we are taught the central dogma of biology. So the central dogma tells us we have DNA. DNA gets transcribed into messenger RNA, which is translated into proteins. All right, bear with me. Proteins are involved in the biochemical pathways in your body. Those biochemical pathways then end up deciding your phenotype or your trait, what you look like. All right, so central dogma, a little bit expanded. What we've talked about and what companies like 23andMe are doing are they're going from the top all the way to the bottom, right? So the very basic level of information and not using all of these intermediate steps where all sorts of things can happen, right? Fortunately, there are all sorts of technologies that are coming up. We've been talking about genomics. There are transcriptomics to measure your expression levels. There's proteomics to measure your proteins. There's metabolomics to measure the metabolites that come out of these biochemical pathways. We love our omics in science. That is completely another lecture and probably best delivered by somebody else. But suffice it to say, there's all of this information that we're now able to gather and to gather in these intermediate steps, okay? So we are indeed still learning. What we have learned to date is vast, right? So tens of thousands of places in your genome that we now know influence your disease risk. And for us to do that, it took millions of people, right? So we needed people to participate in research to be able to help you understand how your genetics affects your risk of disease, right? So I say that somewhat shamelessly, being a data junkie, right? If you get the opportunity to participate in research and you're able, right, you might consider it. You can do this for fun. Like 23andMe gives you the chance to tell them all sorts of things about different diseases. You could participate in research. And you might consider it. You're going to learn a lot. You're going to learn a lot about yourself. You're going to learn a lot about the process. And maybe in a couple more decades, who knows how much more we're going to know, right? So I'm going to thank you. So thank you for participating today to come to learn a little bit more about genetics and how it applies to you. And thank you on behalf of science and a selfish data junkie, should you offer to participate in research in the future. And with that, I would be happy to take questions.
Hi there. Thank you so much for being here. Really appreciate it. I'm a poli-sci major, so I really,
Awesome. Okay.
I mean, this is enlightening. I just wonder, what do you think about CRISPR, Lulu and Nana and Dr. He in
oh my.
out in, I know, I'm just curious, you know, what are your thoughts on that?
Your political science or philosophy? It's interesting. So I'm also in the Human Medical Genomics program, and we had a roundtable where we separated and had discussions about CRISPR and the effects on, and we separated it out. So we talked about, would you be okay with it for your pets? Would you be okay with your kids? Would you be okay if it was to save your kids from a deadly disease that you were carrying? And I will tell you, there was no agreement in the room. So, I mean, my own personal opinion is I think there is so much that we don't know, that how can we change one location and really have an understanding for all of the cascade of things that it might affect. So I'm going to err on the we don't know enough to necessarily be doing that quite yet. But that's just my personal opinion. And I'll tell you, there was not agreement among that room of geneticists. So.
Thank you, Dr. Santorico. This is a really great explanation for all audiences about the complexity of this whole issue. My question for you is, I've seen a couple science journalists submit their DNA to a bunch of different organizations, not just 23andMe, and hear that they come back with different results. Mm-hmm. Now, as a stats professor, how many times should you submit your DNA to different companies? And remembering it's $99 a pop, which is still cheap, how does that add another layer for me to not be so concerned about the results that I get?
I mean, I think the reason to not be concerned is because, so there are a number of reasons why the results differ, right? So there are some technology reasons. Right, so that's another reason why we need to be careful with these until they really are reliably reproduced. And when we look at, so in the forensics case, we currently have 20 places. We have standards for how a person is typed for those 20 places that are tried and true. Right? So we don't yet have that for things like the technologies that 23andMe and Ancestry are using, which means they're going to have some error in them. And that's going to, all of them at this point will have error in them. So until we really have that technology standardized, it's just noise in the system. My guess, and I haven't done this because until recently, 23andMe was the main one that did health, and that was what I wanted, was health. I wasn't looking at genealogy. But Ancestry is now doing that, so now I'm curious. They shouldn't be that vastly different. So I here and there a little bit, but they should not be vastly different.
Thank you, Stephanie.
Sure.
If there's a verified historical increase in the frequency of a disease, does that point towards environmental factors?
It's going to depend. So it could. It could also have to do with gene flow in populations. So some of the extreme examples of slavery, right? So when you put Africans on ships and they're coming over to the U.S. through those horrible conditions, that's going to create changes in the frequency of their alleles. And so there are things that relate to hypertension that we think of as this may be a result of selection from some of those bottlenecks in a population. So it could be environment. It could be changes in gene flow. It could be either or.
Thanks.
Sure.
So how fast do some of these changes in gene flow occur? At the fastest?
At the fastest? Oh my goodness. So I don't know if I can quantify that, but I mean, it's going to depend on how severe, so we call it a bottleneck, right? So if you take a population and you put it under extreme duress to where you have a lot of the alleles dying out, you really can dynamically change that population within a few generations. Fortunately, that's not as much of an issue these days, I'd like to think. So it's probably going to be slower, and so you might think that changes in our lifestyle, whether it's sedentary versus having to do everything for yourself, changes in nutrition, those might create a little bit of a slow change. It's hard to say. So it probably have to be pretty major to have a change over a small span of time.
Hi, if there are many different sequences, which maybe like for hair color, like 11,000 sequences for the one phenotype, how do you assign the relative weight to each of those different sequences?
The relative weight in terms of their frequency or in terms of their impact on disease? Impact, okay. So MC1R, so the red hair gene is a good example because there are lots of variants. So I mentioned, I think 9 that I had looked at. You can look at it either way. So we can estimate and measure what is the risk imposed by each variant. Generally, what people are more interested in is if I put them all together. So how much does that MC1R gene explain? So that 73% that I mentioned, that is describing the effect over all of the different versions that lead to red hair. So there's a couple of things to think about a little further. When it then comes to you, right, you don't really care about those other types. And so you're going to be looking at a different statistic. So you're going to be wanting to know, instead of how much of the risk do all of these things explain, you're instead going to want to know, given my genotype, what's the probability I develop that disease? So it's just a different way of looking at the statistic.
All right, last question to Regina. Thank you, Kelly. So I know people are also very concerned about health insurance companies receiving their genetic information and based on that, making other decisions about health insurance coverage. Can you speak to that a little bit?
Sure. So this is where some of our professional organizations come in. And so it's probably been 5 to 10 years. I don't know, I lose track as I age. But the American Society of Human Genetics lobbied a long time ago to have that protected. So we have GINA, which protects individuals from having insurance companies do that. It could be a problem. So it's one of the things too, if anyone starts to push to repeal some of those protections, we're kind of all going to be in trouble because everyone's data is everywhere now. So I think it's good that we have these professional organizations to lobby for those protections because we do need them for sure. Before we wrap, just to make sure I got all my take-home messages across, I was going to give you a quick wrap-up, right? So we had our questions. Hopefully, if you just woke up, here's the important slide. So part of the story I told you. So over the past 2 decades, we've discovered a huge number of places in our genome that affect your traits, right? So that GWAS catalog I gave you is a great resource for that. Know that the impact of these are relative to you. So I mentioned age, sex, race, the rest of your genome, environment, right? So never look at and treat as the whole truth one specific location. We're now moving to try to combine everything, right? This is science, this is research. We're doing this now. We don't know how well it's going to work. So if you pull out some of those reports, like I showed you for type 2 diabetes, know all those caveats. Scientists themselves don't know yet if they want to believe them. We're still learning. I showed you an example. We're not always right. This is science. It's a process. And participate. So back to the title of the talk. Go out, get cheeky with it, learn a little bit more. And that's a wrap. So thank you for coming.
That was wonderful, Stephanie. We have a bag for you that we've had in Boston. It has PhD 2019 faculty lecture. So,
Nice, thank you. Awesome. And no one will take it.
Yeah, no one will take it. Thank you. So when you were talking about your red hair, and I have a colleague a couple of them over here with red hair, and it reminds me that years ago when our daughter was born and we were looking for a caregiver for her and checking references. And so I talked to this one gentleman and I said, will she take feedback well? And he said to me, lady, God don't make redheads for nothing. And I think that is so true. That, indeed, how special you are. And, how fortunate our students are to learn in classrooms where people who are so knowledgeable and so passionate. And there's something extraordinary when you are able to learn from people who are discoverers and are able to make it so real. So how we're delighted to have you as our first lecturer. Again, the other three finalists, they are representative of a wonderful group of faculty here at CU Denver. And, if I'd heard this, I may not have called, when I was doing my PhD, statistics, sadistics. That's so I was not the math nerd at all. So anyway, this was fabulous, and thank you so much. Would you all please join us on the first floor of this building? There's the Spear Gallery, and there's a lovely reception there. And Kelly, thank you for all of the work that you've done in putting this together.